morning, everyone. My name is Caroline Rodrigues Menezes, and I'm an MD and third-year PhD student at Dr. Krzysztof Polczewski's lab. It is my great pleasure to introduce our very own Dr. Geoffrey W. Abbott, PhD, and the chair of the Department of Physiology and Biophysics at the University of California, Irvine School of Medicine. Dr. Abbott is a professor in the Department of Physiology and Biophysics and the Vice Dean for Basic Science Research and Academic Affairs. He earned his doctorate in biochemistry from the University of London, followed by postdoctoral research at Yale University School of Medicine. He was made a tenured full professor at Weill Cornell Medical College in 2011 and moved to UC Irvine School of Medicine soon after. Dr. Abbott conducts highly meaningful research, having been awarded a $2.5 million 5-year R01 to study modulation of potassium channels, focusing specifically on correcting ectasia, epilepsy, and hereditary spastic paraplegia. State-of-the-art work demonstrated how plant metabolites can be harnessed to prevent seizures. His group was the first known to identify small molecules to rescue episodic ataxia-1 phenotypes, and additionally found how carnosic acid from rosemary is a highly efficient potassium channel opener that inhibits cocaine-seeking behavior in mice. Funded by multiple NIH institutes due to the impactful nature of his work, Dr. Abbott currently studies potassium channel modulation by ancillary subunits, neurotransmitters, synthetic drugs, and herbal medicines. In 2022, his innovative work was awarded a Samueli Scholarship for his molecular mechanistic research. In 2023, he was awarded the Atlee Clark Achievement Award for integrative health research, and deservedly in 2024, Scholar GPS ranked him in the top 1% of all scholars worldwide, and the #1 in the field of potassium channel research for his lab discoveries in the past 5 years of novel and unexpected forms of potassium channel modulation, attesting to the pioneering, leading edge, and significance of his work. Without further ado, please join me in welcoming Dr. Abbott to present his talk today.
Thank you. All right, well, thank you so much, Chris and Grazina, for the invitation to speak today amongst this distinguished panel. Oh, I'm really nervous, slightly terrified, but here goes. So we look for new medicines from plants. I'm going to talk about 5 different disease states today: hypertension, addiction, epilepsy, ataxia, and candidiasis. The major target for the plant metabolites that we look at is the voltage-gated potassium channel. I'm going to talk about other channels as well, but this is the main one. Voltage-gated potassium channels have a voltage sensor here, S1 through S4, which has periodic basic residues in S4, and then they have a pore domain. 4 of these subunits come together to form the tetrameric potassium channel. These potassium channels are important in regulating firing frequency and excitability in excitable cells. The Kv7s or KCNQs I'm going to talk about today are particularly important in deciding whether neurons and other excitable cells fire or not, and how excitable they are. The Kv1s or KCNAs are also important in determining firing frequency and action potential morphology and duration. We're not just interested in understanding the basic physiology, we're also interested in understanding what happens when these channels break. So for instance, in the KCNQ2 or Kv7.2 potassium channel, mutations in these channels cause benign familial neonatal epilepsy and also developmental epileptic encephalopathy, which is a terribly debilitating disorder causing epilepsy and developmental delay. And patients with this are typically wheelchair-bound, nonverbal, and may die actually early in life. So we're trying to understand, I'm trying to impress upon you that plants' metabolites are very important in discovering new medicines, and the potassium channels are actually really important targets of plant metabolites. So first of all, I'm going to go into hypertension. KCNQ5 or Kv7.5 is expressed in the brain. Mutations in it cause intellectual disability and epileptic encephalopathy, but it's also expressed in blood vessels, in vascular endothelial cells. So we were interested in understanding the role of KCNQ5 in potentially modulating vasodilation or vasoconstriction and potentially being a treatment or preventive measure for hypertension. So if you open potassium channels in vascular endothelial smooth muscle cells, such as KCNQ5, you could potentially prevent firing of voltage-dependent calcium channels and cause vasodilation. If you inhibit them, cause vasoconstriction. So we're interested in doing this to potentially treat hypertension. So we asked the question, which may be a bizarre question, Can KCNQ5 opening explain the effects of antihypertensive herbal medicines? For the last million years, us and our ancestors have probably used plants as medicine, and more lately been identifying plants that might have a role in being antihypertensive. So what we did, we took 15 plants, we mashed them up. 10 of them have preclinical or clinical evidence to be vasodilatory or antihypertensive, such as thyme, marjoram, basil, chamomile, lavender, etc. We took 5 controls, such as mint, tarragon, parsley, wheatgrass, and used those as controls. And we extracted all these and then ran them over KCNQ5 channels expressed heterologically in Xenopus laevis oocytes. And we used 2 electrovoltage clamp electrophysiology, which is a 75-year-old technique, to look at the effects of these plant extracts on our KCNQ5 channels. Here's a few just quick examples. So for instance, when we're recording currents, we take the membrane to different potentials from -80 to +40, and then we add our extracts and we do the same thing again. We look at the tail currents where we're normalizing for driving force, so we could look at the voltage dependence. And what we saw with lavender is that it actually shifts the voltage dependence of these currents through KCNQ5 to the left, which means it opens the channels at much more negative potentials. This hyperpolarizes the cells and would be predicted to be vasodilatory if it happened in vascular endothelium. Same thing for fennel seed, which also massively increased the currents through Q5. Same thing for Q-Shen or Sophora flavescens, a traditional Chinese medicine. Now, for our negative controls, such as tarragon or wheatgrass, that don't have any vasodilatory effects, they did nothing to this channel. If you look at all the plants we studied, all of the ones with the folk medicine or other history and preclinical or clinical evidence to be vasodilatory, activated KCNQ5 and hyperpolarized the cell membrane of cells expressing KCNQ5. None of the controls did it. So it's 100% correlation. We think that KCNQ5 activation is a universal unifying factor for the antihypertensive effects of plant medicines. What's the compound? Well, we took one example, Sophora flavescens or Q-Shen, traditional Chinese medicine. We found that allopurinol in an alkaloid in Q-Shen opens KCNQ5. This is the first known KCNQ5 selective channel opener, actually. Metrine and oximetrine did nothing. And then our collaborators in Copenhagen in the Tom Jepp's lab showed that in ex vivo myography studies in rat vessels that actually allopurinol works in a KCNQ-dependent fashion to relax those blood vessels. You can block it slightly with the KCNQ blocker linaperdine. So this kind of proved that it also happens ex vivo. And then also in vivo with a collaborator, a former postdoc from my lab, Zhaoyang Hu, in an ischemia reperfusion injury model, they found that if you do ischemia reperfusion injury by ligating the coronary artery in rats, you can go very quickly into ventricular fibrillation and the rats die. However, if you give them allopurinol beforehand, they come back into sinus rhythm and almost all the rats were saved. So very excited about the potential for these folk medicines and allopurinol in the future. And I'm going to move on quickly. This is a whistle-stop tour through several different diseases. So we're going to go into addiction very briefly now. This is a study that came out a couple of years ago. So in 2023, Ria Manville, a fantastic project scientist in my lab, found that rosemary like we all have in our kitchens, opens up some of the KCNQ channels in the brain, such as KCNQ3 and KCNQ5, and KCNQ3/5 heteromerase. Now, it does not open KCNQ2/3. KCNQ2/3 is the main, probably, channel in the brain forming the muscarinic inhibited M current, which is really important in epilepsy, and I'm going to come back to it. But the rosemary did open KCNQ3 and KCNQ3/5. And more importantly, he identified the molecule in rosemary that does it is carnosic acid, which forms, if you buy rosemary extract from Amazon or some herbal medicine store, about 20% of that rosemary extract is carnosic acid. So carnosic acid is an abiotin diterpenoid. So we thought, well, what can we do with something that doesn't open this main channel that everyone's obsessed about, but opens up Q3 homomers? This carnosic acid is the most efficacious known opener of KCNQ3. It beats all the synthetic compounds out there, shifts the voltage dependence by about -82 millivolts. So it's incredible. But what do you do with this? Well, because most of the channels are thought to form as heteromerase. Well, we have an incredibly talented associate professor, then assistant professor in our department in physiology and biophysics at UCI called Kevin Bayer. I got talking to him serendipitously at a thesis defense, and he said, you know those channels you were always talking about, KCNQs? He said, well, we found something very interesting. In globus pallidus extremus circuits, when we make mice addicted to cocaine, 2 of your channels get downregulated, KCNQ3 and KCNQ5. So I said to him, Kevin, this is a match made in heaven. We're going to try, why don't you put carnosic acid into your mouse model of cocaine addiction and see what happens? Well, it was really incredible, and this was published last year in Neuron. So when you give the mice carnosic acid, it reduces their cocaine-seeking behavior. In this model where they get to either press the lever of saline or press the lever for cocaine, obviously the mice like the cocaine more, but if you give them carnosic acid, it greatly reduces their cocaine-seeking behavior. And he showed through a series of incredible experiments, Kevin is an incredibly talented scientist, he showed that it's via upregulation of KCNQ3 and KCNQ5, which were downregulated in this globus pallidus extremus circuit. So we're hopeful that one day people could be taking rosemary or carnosic acid, maybe preemptively or maybe to help treat their psychoactive drug addiction. So very excited about the future of that. Okay, moving on to epilepsy, and remember I told you about that very important KCNQ2-3 channel. So in folk medicine, and I'm really going through the kitchen herbs now, cilantro, or coriander as we call it in the UK, Coriandrum sativum, there's another plant that's not genetically related called Eryngium fetidum, but they both smell the same. And Eryngium fetidum is actually much stronger smelling and much stronger tasting than cilantro. And they're used in Latin America and the Caribbean as folk medicine anticonvulsants. So we wanted to test the hypothesis that maybe KCNQ2-3 activation is somehow involved. And Eryngium fetidum, one of the common names for it is fitweed, because it's used to stop fits or seizures in children in Latin America and the Caribbean. So long story short, we found that cilantro extract does actually activate the KCNQ2-3 channel. And when we tried the top 9 compounds represented in cilantro extract, we found one, the fatty aldehyde E2-dodecanol, which an EC50 of 60 nanomolar left shifts the voltage dependence of KCNQ2-3 channels. Now, it's quite a specific effect because if you have compounds on either side which have one more or less carbon, they don't do anything. So it's all about E2-dodecanol. So we think this is the molecular basis for the folk medicine usage of Eryngium fetidum and Coriandrum sativum as a folk medicine anticonvulsant in some countries. It binds to a very specific binding site, foot of the voltage sensor of these channels, and also straying close to the binding site for retigabine. Now, retigabine was the first in-class anticonvulsant that works by activating KCNQ2-3 channels, and that's a synthetic compound that's not used anymore, apart from off-label for some of those very severe KCNQ2-DEE children. People are trying to bring it back maybe in lower doses, and we have some ideas about that as well, but it basically, E2-dodecanol binds to a very similar site. And there's the effects of cilantro on the channel, there's the effects of E2-dodecanol on the channel, so very, very similar. All right, and in mice it works too. E2-dodecanol delays seizures. One of the inactive components of cilantro does not, and you can block the effect of E2-dodecanol with a KCNQ blocker. So now we're going to move out of the kitchen. Hopefully I got you ready for lunch. We're going to move out of the kitchen and into the wild. So I am deeply privileged, and members of my lab too, although they may not feel that way all the time, to go out into some of the wonderful wild places in the United States and the Caribbean to collect plants. So we have federal permits to collect in places like Yosemite, Muir Woods. Here's me in Kelso Dunes in Mojave Desert. There's the Salt Lake at Zzyzx in Mojave. This is the beautiful Santa Cruz Island in the Channel Islands off the coast of California. And this is actually Buck Island off the coast of St. Croix in the US Virgin Islands in the Caribbean. So we collect plants from these places, and Boyd Deep Canyon, which is in the UC Reserve System. We collected about 3,000 of these extracts so far in about 48, 49 field trips so far since 2019. We mash them up and we throw them on channels, sometimes doing kind of unbiased screening and sometimes doing very focused screening. And we threw 1,444 of these extracts at 2 different channels, KB1.3 and KB7.23 or KCNQ2-3. Originally, we were looking for the molecular basis for topical folk medicine plants such as witch hazel. So witch hazel has been used for generations. We have always had some in our medicine cabinet at home. And it's both soothing and anti-inflammatory. And in a collaboration with Mike Cahalan and Shiva Othi from the physiology department, we found that witch hazel both inhibits the Kv1.3 T cell potassium channel, thus hindering T cell activation to be anti-inflammatory, while also opening Kv7.23 channels such as those in nociceptive neurons, giving it some analgesic effect. And we found that the compound there was actually tannic acid. And we've had a few papers on tannic acid. Works great as a topical anti-inflammatory and analgesic, but we're not too excited about it as a compound that you might take internally for things like epilepsy. It's not the best thing to be ingesting. So we thought, well, if we can start to look at plants now that activate this to maybe be anticonvulsant but don't inhibit Kv1.3, we'll get away from the tannic acid effect. So that's what we started doing. So we're focusing on some of the plants, not necessarily up here, but more down here, that they don't inhibit Kv1.3, and in particular, Thespesia pulponia, which is the Portia tree, and I collected this off a beach in St. John in the US Virgin Islands. But it's a really interesting tree. It's found in tropical and subtropical coastal regions all over the world. Portia wood, or Thespesia pulponia wood, was used to carve the Ronga Ronga tablets, which are very famous from Rapa Nui or Easter Island. They contain a completely kind of unusual, unknown, just separate language system, which is still being figured out. But also, more importantly for our studies, Thespesia pulponia extract was used in the remote Rotuma dependency north of Fiji as an anticonvulsant, folk medicine anticonvulsant. We didn't know this when we identified Portia tree as being one of the compounds of plants of interest, so this is a really nice link. So if you then apply those plant extracts to the different, to Kv7.2, 7.3 channels, we can see here's one of the ones that inhibits Kv1.3. So this is a hybrid, probably a hybrid between sugar bush and one of the garden escapes from one of the fancy Malibu homes. We collected these in Santa Monica Mountains. So it's like, no one really knows what this is, but it's probably a sugar bush hybrid. We found that this gives us the classic electrophysiological response on KCNQ2-3 of tannic acid. It inhibits at depolarized voltages and activates at hyperpolarized voltages. So we thought we're not going to pursue that one any further. This is four-winged saltbush I picked up from Mojave Desert, Aegioplex canescens. This one has a kind of a weird effect where it keeps activating the channel, but then the IV curve passes through zero. I think this is because the saponins in this plant are punching a hole in the cell membrane and just allowing all ions to come through. And in fact, Native Americans use this as kind of a natural soap because it has the saponins in it. We focused on, however, on Portia tree down here, and we can see that Portia tree provides that left shift that I've been talking about for some of the other compounds. So Thespesia pulponia, Portia tree, is actually a bona fide activator of Kv7.23 without probably tannic acid being involved. And in fact, what we found was that the molecular correlate in Portia tree that opens up these channels is gentesic acid, which is a very interesting compound. Docking predicted it to bind right on top of that, very tightly on top of that tryptophan that's involved in the reticulamine binding site. And mutagenesis studies validated this. It's the most potent known opener. So we discovered from this plant the most potent known opener of Kv7.3 and Kv7.23. So between rosemary and Portia tree, we've discovered both the most efficacious and the most potent openers of Kv7.3. And for 7.3, it's actually subnanomolar. It works in, gentesic acid works in vivo, it delays seizures in mice. Although as you go up in concentration, you start to lose that effect. So we think there's another lower affinity target. And here's a picture of Rotuma dependency. And I just feel like I had to get out there just to explore this further. Nothing to do with the quality of the beaches or anything, but it looks beautiful. So, all right, so the last two disease states I'm going to talk about don't involve KCNQ channels. Ataxia involves those KV1 channels, which are still voltage-gated potassium channels. So ataxia is a collection of movement disorders, genetically kind of heterogeneous. We're going to talk about episodic ataxia 1, which is caused by mutations in the KV1.1 or KCNA1 gene. And ataxia can come along with heart problems, slurred speech, vision problems, et cetera, et cetera. So we were working on ataxia here and there. We collaborate with Rich Sidler, who's a pediatric neurologist, to characterize a Kv1.1 mutation, L155P, that was in a mother and daughter who'd been misdiagnosed with epilepsy. Turns out they had ataxia. It was the first known ataxia mutant in the N-terminus of the channel, the cytosolic portion. Turns out if you measure the currents, there's a loss of function. Patients are typically heterozygous. If you knock out both copies and mimic that, then you get zero current at all. So, okay, we characterized a couple of these, but there was no known drugs that directly fix the channel function in ataxia. So the patients are treated with things like carbamazepine, which inhibits sodium channels, but doesn't directly fix the voltage-gated potassium channel problem. So we had a study in 2023 where we learned from the Kwakwaka'wakwa First Nations in the Pacific Northwest that used bladderwrack kelp, Pacific 9-bark, and nettles, those extracts, to treat ataxia. And what we found was that there were compounds in those plants that actually do fix 2 of the 5 different ataxia mutants that we tested. So this is wonderful, and it was the first example of small molecules that we found, such as gallic acid in those plants, to directly fix mutant Kv1.1 potassium channel function in ataxia. But 2 out of 5 isn't that great, so we went further. So Native Americans have a history of using pine tree extracts for movement disorders, paralysis, this type of hyperexcitability type of disorder. So we learned from them, and we did a kind of a targeted study of different conifer trees. And what we found was that actually they're really good at opening, this is wild type Kv1.1 channels, shifting their voltage dependence to the left. The only one that doesn't, strikingly, Pinus albicaulis, is the only pine tree that we could not find any evidence of folk medicine usage in Native American culture. So it's pretty interesting. This is the only one that doesn't do anything to Kv1.1, apart from some inhibition, but it doesn't shift in the way we would hope. So in the end, what we did is we focused on actually the saguaro cypress because of its overall profile, and discovered that there's a metabolite in there called pisciferric acid, which the plant is named after, C. piscifera, which is an abiotin diterpenoid similar to carnosic acid from rosemary. When we applied pisciferric acid, or metabolite Z, as I'm calling it here, to 12 different mutants that cause episodic ataxia, we found that it was universally rescuing all of them. It didn't always rescue full wild-type function, but it restored at least some function. And a good example here is R307C. The wild type is in purple here, the wild type voltage dependence. The mutant is in black, and then you can restore completely wild type voltage dependence. So really exciting. Pisciferric acid at 12 micromolar, which is around the EC50, increases activity through all these mutants and left shifts or negative shifts of voltage dependence in all those mutants. So this was incredibly striking result, I think, and it says that pisciferric acid could be a potential therapeutic or lead to a potential therapeutic. We also had a paper that just came out in August showing that pisciferric acid also rescues 19 out of 19 Kv1.2 mutants that we tested. So 1.2 forms heterozygous complexes with Kv1.1, but 1.2 is associated with epilepsy, ataxia, and hereditary spastic paraplegia. So we're really excited about the therapeutic possibilities of pisciferric acid. Does it work in vivo? Well, yes, it does. So when we put these mice on a rotarod, the mutants fall off much more quickly. When we give them pisciferric acid, they don't. They act like wild type. When we look at a balance beam, you have to stress the mice more before you do the balance beam to kind of recapitulate what happens in human episodic ataxia, where people get stressed before they get the ataxia. And maybe you could ask them to give a talk in front of all these people, for instance. So on the balance beam, unstressed, the mutants are just like the wild type. However, if you then have a blinded scorer counting hindfoot missteps, you can see that the mutants have more than double the amount of missteps of the wild type, which is consistent with ataxia. When you give them pisciferric acid, just 1 milligram per kilogram IP, it goes away. So pisciferric acid works in vivo in mice. And you don't have to believe me, you can see what's happening. So these are mice on a balance beam. These are isoproterenol treated, but not treated with pisciferric acid. The wild type does fine. The mutant keeps falling off, but also, more importantly, you keep seeing these hindfoot slips. It just can't balance as well. And they're counted by a scorer, an undergraduate named Amy Lee, who didn't know the genotype or the treatment of these mice. So then we move on to the pisciferric acid-treated mice, wild type again on the left. And we can see that wild type's doing just fine, as one might expect. And now the mutant mice don't have nearly as many missteps. So pisciferric acid works in vivo to fix the symptoms of ataxia, and hopefully one day pisciferric acid or a compound like it will be used in people. And actually, we've now got a load of more experiments showing that carnosic acid, which is in rosemary extract, and I take rosemary extract every day, for instance, at the levels that are in the extracts and supplements, does the same thing, rescuing ataxia in mice. So carnosic is an amazing molecule. How does it work? Well, another very talented physiology and biophysics assistant professor, Alvin Yu, who does MD simulations, showed that you can just sprinkle pisciferric acid in extracellular space, And what happens was it finds its way to the voltage sensor binding site. It makes the voltage sensor more compact. It makes it move through electric field much more easily in the channel. And that overcomes the effects of the mutants. So fantastic. Finally, the final part, bit of a kind of a different direction, candidiasis. So now we're not talking about voltage gate channels anymore. So the voltage gate channels have the voltage sensor, they have the pore helix, and they have the pore domain. Inward rectifiers don't have the voltage sensing domain. K2P channels form as dimers, and they have no voltage sensor. And then TOC channels are from fungus, and they have these 4 transmembrane domains and then the 2 pores, and they form as dimers. Now, Steve Goldstein, who's our Vice Chancellor of Health Affairs, first discovered these in 1995, and that was the first known K2P channel that works as a dimer. So we're interested in asking whether we can target these channels to be as an antifungal target. So Ria Manville in my lab figured out a way to AI screen plant extracts to look at folk medicine history to find out examples where people had used plants as antifungals. Then he tested the 29 hits from that screen and found thyme, oregano, cinnamon, and a plant called Terminalia modulated the TOC potassium channel in vitro. And thymol was the compound that he found in thyme that was inhibiting the TOC potassium channel. So we also found, using growth assays and plate assays, when we did a structure-activity relationship for compounds related to thymol, TBMP, a compound we're calling TBMP, was able to kill fungus and also inhibit the TOC potassium channel. And the structure-activity relationship shows that killing of the fungus and inhibition of the TOC potassium channel actually kind of goes somewhat hand in hand. Importantly, TBMP was also able to inhibit fluconazole-resistant and kill fluconazole-resistant fungus. So we now have a structure of the potassium channel that Steve discovered 30 years ago in combination with Alexandra Miller, who runs a cryo-EM lab and is a very talented assistant professor in the University of Iowa, and a grad student, Bryce. We can see that, just like Steve predicted, it's a dimer. Unlike human K2P channels, it lacks the cap at the top, and it has an extra 4 helices to the side, just as Steve predicted. We're calling these extra 4 helices a talc domain for the TOC auxiliary subunit-like channel domain. And they, while having low sequence homology, or 0 hardly sequence homology, with a certain group of proteins, they have 100% almost identical structural overlap with TARP domain proteins, which are auxiliary subunits from channels in the human nervous system, such as the voltage gate calcium channels and AMPA receptors. So by some quirk of evolution, they have exactly the same fold as the auxiliary subunit. This one's covalently on the side of the TOC channel. So there it is from the side, and then we remove the talc domain. That's what it looks like. The other unusual feature, apart from having no cap up here compared to the human channels, is they have the C-terminal bundle down here. We don't know quite what it's doing, but we know that the talc domain, we published a paper actually, and Steve was on that earlier last year, this detects changes in pH, which is super important for Candida to be able to populate the human gut. So we think that this is a kind of like not a voltage sensor, but an external stimulus sensor. How do the channels gate? Well, tetrameric potassium channels gate by having a bundle crossing that then opens. Human K2P channels have a much more subtle situation where one of the transmembrane helices moves up. When it moves up, it exposes, or it shuts off this window in the side of the channel, this kind of lateral fenestration, and that's thought to keep lipids out and let potassium go through. In the TOC potassium channel from Candida albicans, we see a similar kind of event happening. There's a 25-degree twist in this helix, and if you look from the side, you see exactly the same thing as you do in the human K2P channels. The fenestration closes, and the channel is probably opening and allowing potassium to come through. Now, we also, thanks to Alexandra Miller's great work in her lab, we now have a higher resolution structure of the channel with TBMP bound. It does exactly the same thing as we see in the down state, the closed state, without the presence of the drug. So TBMP sits next to that helix, twist locks the helix to close the channel, And that's how we think TBAP inhibits the channel. So in summary, I hope I managed to persuade you that plants are a treasure trove of compounds, and they act largely, there's lots of effects on channels, voltage-gated potassium channels, and fungal potassium channels. And we think that these are a source of potentially novel therapeutics in the future. Just to show you some snaps from our trips, we see some amazing animals along the way. And here's Rian at 4 o'clock in the morning collecting plants on Santa Rosa Island, wondering why he's not at the rig doing experiments. He's an amazing project scientist in the lab, and you see his name comes up again and again. He's first author on many of the papers I've shown. That's a Channel Island fox, which weighs about 5 pounds and only lives in the Channel Islands off the coast of California. Oops, that's a Mojave desert tortoise down there on the left, and that's a tarantula that wandered through our camp right before we were going to go to bed in Mojave. So other cool places we get to go. This is me collecting a small plant very carefully from the Kilauea volcano earlier this year, putting it away in my bag nicely. Then on day 2, this started happening. Day 3 started getting a bit more violent. And then on day 4, we had a full-blown 300-foot eruption. So I don't think it was my fault, but moving on quickly. So the next frontier is I was out in August in Renomafana in Madagascar, sorting out collaboration to go back there next year and collect plants. Luckily, after 24 hours of flights, I didn't have to take an oxen and cart to get to the rainforest, or indeed one of these things. The excellent driver we had, Fabrice, took me on a 14-hour pickup truck ride. You really get to know people really well on a 14-hour pickup truck ride. And that's the view from my window at Centre Valbio Research Station in the middle of the jungle in Madagascar. I got to film this guy super close up. He sneezed on me a couple of times. I came down with this terrible, shivery, achy stuff after 36 hours of that happening. I was convinced I was going to be patient 0, but it passed, and I got home safe. But just looking at the natural world around us, and we become especially in tune to it when we go on these trips. We see things like this moss gecko on this plant here, which is incredibly well camouflaged. We see beautiful plants, and we see the beautiful lemurs. And it reinforces the fact that, we're looking for new drugs from plants, but we're also trying to put a molecular basis and a credibility and validation in some cases to traditional cultures and traditional folk medicines who came before us. They discovered all this stuff. We're just showing people how it works. And it just reinforces the importance of protecting these natural habitats for the future. So the golden bamboo lemur, and I'm going to go back to show him again if I can. The golden bamboo lemur, who sneezed on me. So in 1986, the golden bamboo lemur was discovered. There's probably only around 1,000 of them left on the planet. They all live in Renomafana. Because of that, the government in Madagascar, together with scientists in the US and Madagascar, preserved that rainforest. The rest of the area is highly deforested, but they realized the importance of protecting the species and all the incredible stuff in that forest. So we just hope that we get back on track in terms of realizing what an incredible treasure trove the natural world is around us and protecting these amazing creatures and plants before they go away forever. So I'd like to acknowledge all the people that helped. Rian's been incredible. He's just a fantastic scientist in my lab. Kevin, Alvin, Lorenzo in Alvin's lab, Shiva, Angel, Mike Cahalan, incredible colleagues in physiology and biophysics, Tom Jeps from Copenhagen, Rich Sidlow, Alex Miller, who her lab did all the cryo-EM that I was so happy to talk about today. Thank you for all the funding. I want to highlight especially Susan Samuel Integrative Health Institute right here at UCI. Thank you to the National Park and UC Reserve System for allowing us to collect plants in these wonderful places. And thanks to the Daily Mail newspaper for highlighting my lab's work and getting me a new nickname. So really appreciate your time today. Thank you.
Jeff, that was a tour de force, to say the least. Just wondering, for the candidate, the last part of that story, that sort of came out the blue. I wasn't expecting that. Excuse me. Have you tried any of these medicines in vivo, in animal models or anything?
Not yet. So we're still TBMP, full disclosure. I don't think it's ever going to be an antifungal because it's kind of has toxicity issues as well. Although you could argue it might be good for killing things on surfaces. But we want to look at Steve Goldstein's work on TOC. he followed up the original cloning paper, which was the first K2P of any kind, and then other people followed. But he followed it up showing that yeast killer toxin opens up other tox channels in other yeast, kills the yeast by opening them. So we're interested in going back and looking at that Terminalia plant that I showed in the screen to see, because that's two different extracts of the same species collected from two different places, and they both opened it. So I really believe that result. So we're interested in finding out the compound in Terminalia that opens TOC, because that actually might be a more viable antifungal than the inhibitors. We're still interested in inhibitors, and also it gave us that nice gating kind of nuance about how the gating works in the channel. But we're interested in looking at the openers, and then absolutely we can collaborate. You do work in this area, collaborate to look at in vivo stuff. But so far, just in vitro.
Thanks.
It's nice to see how much fun you have in lab. My question has to do a little bit more with the pharmacokinetics. So like how much rosemary do we have to eat to get enough concentration in our blood that'll be effective?
Right.
And also how long does it last? Do we have to have like rosemary all the time?
Right, that's a really good question. So how long does that, what's the pharmacokinetics or pharmacodynamics? So the first answer is we have a grant to study that, and we're doing it right now from the Ataxia Foundation and also from NIH. So the amounts, judging, if you kind of extrapolate from the mouse studies and you kind of divide by 7, I think it is, which is kind of the rough calculation of how much humans need compared to the mouse per kilogram, the amount I take in my rosemary supplement every morning would be enough. So how often one might have to take that, I don't know, but that's easily enough, predicted to be easily enough. Carnosic acid gets through the blood-brain barrier. We've shown that pisciferric acid gets through the blood-brain barrier too, but also these things might work on peripheral nerves as well, where Kv1.1 and 1.2 are expressed, but they definitely get through the blood-brain barrier. So the good news is that carnosic and pisciferric, especially carnosic has been done to death, is really, really safe. You can add huge amounts before you have any toxicity problems. So that's ongoing studies funded by National Ataxia Foundation to figure out exactly how much, yeah.
Okay, so this was an amazing talk, obviously, but just to flip the question a little bit, did you find all these modulators of potassium channels because you work on potassium channels, or are they doing something really critical in plants that could guide your search in the future? That's one question. And question number 2 is, have you worked with any phylogenists of human genomes to see if there are any correlates in the regions where these are highly used with the evolution in humans of potassium channels?
So first question, so second question, no. First question, so, you could say I've been studying potassium channels for 30 years, so we're interested in this. We know they might be doing other things to other targets as well, although I showed you examples where we block the effect with a KCNQ specific blocker, so that's a fairly good evidence. But there could be, we just focus on potassium channels because we're interested in them. They're highly influential in excitability in excitable cells, and so it makes sense to target them for hyperexcitability disorders, and also specifically where we're fixing a particular mutant of a potassium channel, it makes sense. But the question often arises, why are these compounds so good at opening potassium channels? So there's a few theories. Insect feeding behavior might be dictated by, there's some channels in insects which are quite similar to the human ones, and so it might be a way for the plant to regulate insect feeding behavior and stop being eaten so much. There could be there are plant potassium channels that might use those compounds for signaling, for instance, so there's a couple of examples. And then the other thing could be that this is just coincidence. I mean, GABA is present in bacteria and plants. It's not there as a neurotransmitter, but then all of a sudden, as evolution often does, it's been co-opted to be a neurotransmitter. So it could just be coincidental. We often get told that we have a really high hit rate. People make jokes about me just wanting to go to the beach all the time, which may be partially true, but we have a really high hit rate compared to these huge, compound screens. And it's because our compounds in plants have already overcome the biocompatibility hurdle. They're in living cells already, so we're getting rid of a lot of stuff that is not compatible. But there may be more to it than that, as you alluded to. We may be uncovering signaling networks that happen in plants or between plants and their grazer, herbivores, that we don't fully understand yet. Yeah.
Thank you so much. It was
Thank you.
wonderful.